Yeah, first of all, thanks a lot to all the organizers for inviting me and to have the opportunity to be still part of the team here in Zurich, even though I'm physically not in Zurich anymore. And yeah, so the aim of this very first introductory lecture here of the course is to discuss with you the most important basic principles of targeted proteomics in general and of selected direction monitoring in particular. And as we have just seen from this raising your hands, I think we're here a very heterogeneous group of people. There are some people already very experienced with SRM and some are beginners. So I already would like to apologize at the very beginning to those people who have maybe already have a lot of background in SRM and where this introductory lecture will be more of just a repetition of things they already know. But I still hope that you can all benefit from yet another wrap-up about SRM. Okay, and I have structured my presentation into three main parts. The first, and each part is headlined by a fundamental question regarding SRM. So in the first part, we will discuss a bit what SRM actually is, how it works, and why it's something useful to do. Then in the second part, we will design a first targeted proteomic experiment together and see what's important to consider. And then in the last part, we will just look at some real data that you get an idea of how the data looks like. Yes?
Just a quick question, will the slides be available?
Of course, so all the slides will be downloadable and all the videos will go on YouTube. You will have, I think you...
There's a link actually on your USB stick which links to a Google Drive where the slide will be, but there's nothing in there yet, so but that's where they will be.
And as you already did, please, whenever you have questions, just interrupt me at any time. Don't hesitate. You can ask your questions at any time. Good, so let's start with the first part. What is SRM, how does it work, and why is it useful? So in the field of targeted proteomics in our days, this term no longer refers to only a single mass spectrometric technique. But actually by now there are several techniques that we call techniques of targeted proteomics. Definitely the most established and therefore most frequently used technique in the field is selected direction monitoring, SRM. But especially in recent years, by using high mass accuracy, high resolution mass spectrometers, also new approaches have been developed, like parallel direction monitoring or swath MS. And while today, on this first day, we will mainly focus on SRM, you will also hear about the other two techniques a lot throughout the rest of the week and get introductory lectures and do tutorials also on those. One complicating thing in this field of targeted proteomics is the fact that for these three different techniques, various different names or nomenclatures exist. And people who are new in the field find this often very confusing, and people who are a long time in the field find this confusing too. So for example, for the term SRM, you also sometimes find the expression multiple direction monitoring in the literature, and both of these techniques refer to exactly the same technology. It's the same thing, it's just a different name. The same holds true for PRM, so sometimes you find targeted MS-MS, MRM high resolution, MRM-HR, or people also call it pseudo-SRM or pseudo-MRM. All these different names refer to exactly the same technology. And also for swath MS, very often now people use the term data-independent acquisition, DIA, or also hyperreaction monitoring, HRM, was coined in the field. And this is, of course, not great, and it's confusing, but the major reason why we have all these different naming conventions is because different companies of different mass spectrometers trademark for their instrument a specific name, and then it cannot be used easily by the whole community. So also in the future, I'm afraid we have to live with these different namings referring to the same technology. And therefore, it's very important that you're aware of this and that you now know the differences and the commonalities between all these names. Okay, so, but what's very easy to remember is that no matter with which type of targeted proteomics you're working, the benefit of all of them is always the same. So, and what's this benefit? Well, if you have a project where the overall aim is to globally map the proteome in your sample. So if the aim of the project is to identify as many proteins and PTMs as possible in your sample, then shotgun proteomics or discovery proteomics is the method of choice for you. However, when you focus less on identification but more on accurate and precise quantification, then targeted proteomics becomes an interesting technique for you because you can achieve better quantitative results. Your quantitative results will be more consistent and more reproducible than what you can usually achieve with shotgun or discovery proteomics. And that's the case no matter whether you work with SRM, PRM, or SWOT, all should deliver better reproducibility. And the fact that these advantages of targeted proteomics are more and more acknowledged in the field and recognized, you can see that it got selected as Method of the Year in 2012 by Nature Methods. Okay, so now let's go a little bit more into detail how SRM really works. And what you see here, you have probably seen in many presentations of proteomics before. It's what I call the standard bottom-up proteomic workflow. In all our projects, we usually start with some kind of sample that contains proteins, and the workflow is always to first extract the proteins, to digest them into peptides. Then these complicated peptide mixtures need to get separated, usually at least by doing an HPLC step. And then the peptides that elute from your chromatographic column here need to get ionized before they enter the mass spectrometer. And until this point here, this workflow holds true for any type of bottom-up proteomic experiment, no matter whether you're discovery or targeted proteomics. The difference only arises at the very last step of the workflow, namely here at the step of the mass spectrometer. And if we want to do an SRM experiment, the mass spectrometer we work with is a triple quadrupole mass spectrometer. And as this name already suggests, triple quadrupole mass spectrometers work with 3 quadrupole mass analyzers that are used in a row. The first and the third quadrupole both serve as mass filters, and in the second quadrupole, the peptides get fragmented, so that's the collision cell. So if now you run an SRM experiment, at a certain time point, a mixture of different co-eluting peptides might elute from your column, and then this mixture of peptides will enter the first quadrupole, and ideally now just one out of these mixture of peptides will get selected and will be able to pass the quadrupole. This peptide precursor will then get fragmented in the second quadrupole, and in the third quadrupole, again, just one out of this mixture of fragment ions will get selected, will be able to pass the quadrupole, and what you record then in the end is the intensity of this fragment ion over time. This is what we call the SRM trace, and it's this trace that you will use to identify and also to quantify your peptides and proteins with SRM. So the unity that you always measure, or where everything is about in SRM, is always a pair of a precursor peptide ion that you select in Q1 and the corresponding fragment ion that you collect or that you select in Q3. So we are always talking about Q1, Q3 pairs. And there is a scientific expression for these Q1, Q3 pairs. They are called transitions. And you will hear this word very often throughout the rest of the whole week. So it's very important that you know a transition in the context of targeted proteomics refers to a precursor ion and the corresponding fragment ion. So what are the advantages of working with triple quadrupoles and doing such an SRM experiment? So one advantage of the triple quadrupole is that you have these 2 filtering steps. You are filtering for a specific ion twice, and this is why you can quite efficiently reduce the noise in your measurement. And this is why with SRM you can measure quite sensitive different analytes in your sample that are present in your sample at quite different concentrations. Then a quadrupole is a very good mass analyzer in terms of transmission efficiency. So 95% of your peptide precursor or your fragment here will actually be able to pass this quadrupole. And it's again a reason why you can measure quite sensitively. And the third advantage is that you can rapidly switch between these settings here in Q1 and Q3. You can select different precursors and fragment ions, and this is why you can do this multiplexing, so you can not only measure a single transition, you can measure many transitions in one SRM injection. There are also, of course, disadvantages. The major disadvantage of working with a triple quadrupole is its low resolution. So especially compared to the high mass accuracy, high resolution instruments that are out there, the Orbitrap or the TOF analyzers, the resolution that you can achieve here, though this precursor isolation window is usually in the range of 1 Dalton, 1 m/z. Which is very poor compared to the others. And there is this second disadvantage of triple quads or of SRM in general, and this disadvantage is that you need to know precisely what to look for in advance, which means you need to tell the instruments these filters that it should select for. If you don't know these numbers, if you don't have these parameters, you cannot perform an SRM experiment. Okay. So let's have a look at a specific example, and then I think it will become very clear. So let's assume this is our target peptide that we want to measure with SRM. You can see already indicated here the Y and B ion fragment ion series. And if we now want to measure this peptide with SRM, what we need to give to the instrument is a table with parameters. This table contains various columns, and these columns that are important for every SR measurement is first of all this column of Q1. So this column contains the mass to charge ratio of our target peptide, this precise value. And as different peptides can have different charge states, so they can be doubly, triply, quadruply charged, this value can change depending on the charge state that the peptide has in your measurement. So you already need to know what's the major charge state of my peptide in my measurement. It's not just enough to know the sequence and the theoretical monoisotopic mass, but it's also important to know the major charge state or the dominant charge state. Same holds true for this Q3 column here. So there you need to select now the fragment ions that you want to measure for this peptide. We have selected here Y5, 7, 8, and 10. And again, you need to know the mass to charge ratio of these fragment ions. And then there are two additional columns. One is called collision energy. So that's the energy that you want to apply in the second quadrupole to fragment your peptide. And the last column is the dwell time. So sometimes dwell time is also called accumulation time or acquisition time. What it means, what all these expressions mean is that's the time that you want to give the instrument to accumulate signal for a specific transition over time. And usually the longer you give the instrument time to accumulate signal, the better the instrument can differentiate a true signal from the noise. So ideally, you want to have long dwell times that you get very clean measurements and that your noise can be very low and your true signal really increases over this noise. Okay, so what happens once you have generated this table, you've copy-pasted it usually into your triple quad, you press the start button, and what the instrument will do now, it will go to the first line that you provide. It will set Q1 and Q3 according to the two values that you defined, and then it will accumulate the intensity of this specific fragment for 250 milliseconds, and then record an intensity value that you get after 250 milliseconds. Once this value is stored, it will switch to the second line of the table and do the same. So in this example, Q1 stays the same, but we are looking at a different fragment ion. It will accumulate the signal over 250 milliseconds, record a second intensity. And like this, the instrument will cycle through the whole table that you provide. And once it reaches the last line of the table, it will simply switch back to the first line and continue cycling through this table over and over and over again, over the whole HPLC gradient that you apply in your LC-MS experiment. And if you're lucky, and if you have a good, nice target peptide, and if you did everything correctly, the data that you will get from your software analysis might look like this. So here you see the 4 different transitions indicated in 4 different colors that we have measured. And one thing that you can see immediately is that all those 4 transitions have the same elution time, so the peak apex is at the same time. And they also all have exactly the same shape. And that's already a first very important prerequisite. For transitions that are coming from the same peptide, they all should have exactly the same chromatography, because the chromatography is taking place on the peptide level, right? There the peptide is still together, not fragmented. So all the fragments that come from the same peptide should show exactly the same chromatography. However, what you do see is that these different transitions have different intensities. And these different intensities reflect the MS2 fragmentation pattern that we know also from our shotgun experiments. Different fragments are formed with different intensities, and that's also exactly what you see here in these peak heights or peak areas.
Excuse me.
Yeah?
How do I select which fragments
Mhm.
I search in traces?
You will talk about this a lot.
So can you repeat the question?
So the question was, how do I select those 4 fragments? And that's, of course, a very good question, and we will dedicate the whole second section of the presentation just on that. We will come back. Okay, so, but what I want to clarify here is that these smooth lines that you see here are actually not what the instrument really measures. This is already these extrapolated lines coming from the software that you use to analyze the data. What the instrument really measured, as I just showed you before, are distinct intensity values over time. So you just get a distinct intensity for a certain transition at a certain time point. So you get just single measurement points, and the software makes out of it a nice and smooth curve. Now, one very important additional parameter that you need to consider in SRM, but as well in PRM, and as well in SWOT, and actually as well in shotgun MS1 based quantification, is the time difference between 2 of these neighboring points. And this parameter is called the cycle time. The cycle time is defined in SRM as the sum of all the dwell times that you have in your transition table, in your table list. So in this little artificial example, we have measured here 4 different transitions with a dwell time of 250 milliseconds. When you sum them all up, this ends up at 1 second, which means that the time difference between 2 of these neighboring points is 1 second, because the instrument needs 1 second to once cycle through this list and to come back and to measure the same transition again. Is that clear? So the sum of all the dwell times is the cycle time, and the cycle time defines how quickly I measure points over this elution profile. And that's a very important point to think about. And how the dwell time and the cycle time are actually influencing the peak quality that we are measuring is shown here once on one example. This figure is taken from a review from 2008, and I think it very nicely shows the importance here. So what you see here in black is a theoretical chromatographic trace. In our case, this would be an SRM trace, but could be also any other chromatography-based proteomic measurement. Now, this peak was recorded with different dwell and different cycle times. And then you can see what happens to the peak when you use that. So let's first look at the purple and at the red line. So these 2 lines here were recorded with very long dwell times. So that's good. You give the instrument a lot of time to differentiate noise from background. But also the cycle time was low. So in the pink case, it was 20 seconds. So in this case, we have these black curves elutes over roughly 30 seconds. So 30 seconds is the peak width. If we have a cycle time of 20 seconds, this means we will only get 2 data points over this elution profile recorded in our measurement. And you can see immediately with only 2 data points, these 2 data points, no software algorithm in the world will be able to really extrapolate the true peak shape. You will get something that will be by far too low compared to the true peak. Even when you halve the cycle time to 10 seconds, it's the red curve, you end up with maybe 3 or 4 data points. Still a very poor peak quality. This will lead to a very poor peak quality. And only when you're here in the range of 2 seconds, that's the blue and the green curve, you actually get very close to the true peak, to the true quantitative value from your measurement. And the difference between blue and green is just the dwell time. So in blue, you have a very short dwell time of only 5 milliseconds, and you can see the peak is much more spiky. That's just because you have more noise there. The time was very short for the instrument to accumulate the signal, so the peak is much more spiky than with 50 milliseconds, where you really end up with a nice chromatographic peak shape. Okay, so with this figure, I think it becomes very clear that for every SRM measurement, you need to perform a balancing act between 3 parameters: the dwell time, the cycle time, and the number of transition and peptides and proteins that you want to do in one SRM measurement. And the goal that you always want to achieve in all your measurements is you want to be as sensitive as possible. You also want to measure very low abundant proteins or peptides in your sample. And if you want to be very sensitive, the dwell time, the time you give the instrument for a specific transition, should be long, because then you can nicely detect small peaks above the noise. And the rule of thumb, what people often apply when they work with triple quadrupole mass spectrometers, is that this dwell time is somewhere in the range between 5 milliseconds and 100 milliseconds. So if you're interested in a protein or peptides that are quite high abundant, it's maybe already sufficient to just use 5 milliseconds dwell time. You will already get a decent peak. If you're interested in proteins that are very low abundant in your sample, you must use dwell times rather in the range of 100 milliseconds, maybe even higher. Another goal that you want to achieve, you always want to be accurate in your quantitative measurements. And for that, you need short cycle times. You want to have many data points over your elution profile to nicely reconstruct the true peak shape. And here, the rule of thumb that people try to achieve is to have at least 8 to 10 data points over your elution profile. And now you can, of course, understand that depending on your peak width, this will lead to different cycle times. So if you are working with an LC setup that has a very long gradient, or that has a specific resin material, maybe you have average peak width in the range of 1 minute, and then you can have a much longer cycle time than if you work with an LC setup where the peak width is very narrow, and then you must more quickly cycle through your transition list and use shorter cycle times. So this is something where actually it is super important to know your own LC setup and to figure out what's the average peak width in your LC setup, and then accordingly you must define your maximally allowed cycle time. And very often with standard nano HPLC systems many people work with, you need to achieve cycle times in the range of 1.5 to 4 seconds. That's often the case, but really this is something you need to figure out yourself on your system you're measuring in an initial screen or initial survey analysis. And then there is this last parameter of number of transitions. So again, usually people are not aiming just for measuring a single peptide or a single protein, but you want to quantify several peptides and proteins. And here the rule of thumb, what people often work with, is to target each protein not just with a single peptide, but always try to use several peptides per protein. 3 to 5 is what often people aim for. And each peptide, again, should be not measured with just a single transition, but with several transitions, 4 to 6, depending on your specific project. And the maximum number of transitions that you can put into a single SRM run, so the maximum number of lines that you can have in this table that I showed you, is in the range of 1,000. 1,000 transitions you can add. But even this 1,000 transitions you can only achieve with a specific trick, and this trick is called retention time scheduling. Does anyone of you know what retention time scheduling means? Can somebody explain it? Maybe with a microphone. Sorry.
Who was it?
Yeah.
Scheduled means you are doing MRM in a retention time window.
Exactly. So when you do scheduled SRM, you no longer monitor transitions over the whole gradient that you look for, but you just focus on a specific window over your gradient where you expect your peptide to elute. So in this example, we are aiming for measuring 4 peptides with SRM, and we already know that peptide 1 elutes first, and then second, and then this third, and this fourth. So what you do now, when you try to measure these, you would measure the transitions of peptide 1 only from 8 to 18 minutes. And then you would stop measuring those, because you expect your peptide somewhere in this range to elute from your column. And peptide 2, you would just measure from 18 to 30 minutes, and so on and so forth. And like this, you decrease the number of transitions that you need to cycle through in each segment, and thereby can, for example, improve on cycle time and dwell time, or you can add more peptides, and so on and so forth. You will hear a lot more about this retention time scheduling also in this afternoon from Ariel, and how you do this, but it can help. It's important that you understand it can help to increase the number of measurable transitions per run by a factor of 5 to 20. So these 1,000 transitions, this thing that I told you, is where you really work with scheduled SRM and where you work with windows that are quite narrow. Because of course, the more narrow you make the segment around your peptide that you expect, the bigger the benefit of the scheduling trick is. But at the same time, when you make it too narrow and when you have some variation in your HPLC, it might be that your peak shifts out of the window completely, and then you will get a wrong measurement as well. So it's a trade-off that you need to do. And of course, this whole scheduling thing you can only perform when you have accurate retention time information available. When you don't know where your peptides dilute, you cannot apply it. Okay, so just to summarize this first part, the performance profile of SRM is shown here in green, the advantages. So SRM is always the ideal technique for you if you want to monitor a small set of predefined target proteins or predefined PTMs, and if you want to do this over many samples. A major advantage of working with SRM and working with these triple quadrupole mass spectrometers is that you can span a quite large dynamic quantification range. So 4 to 5 orders of magnitude is what you can measure in a single experiment. So you can in one shot measure a peptide that is 5 times, up to 5 times less abundant than the highest concentrated peptide. And this is one of the largest dynamic range that you can span compared to other mass spectrometric techniques. SRM is very reproducible and repeatable, so it's again this major advantage of SRM, and you will see, I think, a lot of data throughout this week that shows this. And SRM is, as I said in the beginning, the most established technique of the targeted techniques, so it's well supported already by software tools. You will see Skyline throughout this week, and there are a lot of spectral repositories out there where you can which you can use as starting points for generating targeted assays and so on and so forth. So you get a lot of great software support and repository support. The disadvantage we already discussed. So you can only perform an SRM measurement when you already know which are your target proteins, which are your target PTMs. And this is what we call, you need to have an SRM assay available for every target that you want to monitor with SRM. And now in the second part of the presentation, we will discuss what this SRM assay looks like and what this exactly is and how you pick transitions and peptides. Then there is a disadvantage of the low resolution of the triple quads, and this causes more interferences, so you will have more problems with interfering peptides and contaminants in your sample than, for example, compared to using parallel reaction monitoring. And this will be also be a topic throughout the rest of the week. And you have this limited multiplexing capacity, so roughly 1,000 transitions per run is what you can do maximally, and especially in the light of these last shortcoming, these other approaches like PRM and especially SWOTH-MS have been really pushed in the past years to overcome this last limitation and to do more large-scale analysis. Okay, there's one more last analogy I want to make for this first part, which I think once more demonstrates the differences between different mass spectrometric techniques by using a very easy to memorize and very easy to understand example or analogy, and that's the example to exploring a new city. So let's assume you would like to explore Zurich for the first time in your life, and then the shotgun way of exploring Zurich would be to fly into Zurich, to have a look at the skyline, and to then visit during your stay in Zurich all the tallest buildings in the city. And by taking this approach, you would hope that these tallest buildings in the city are actually the most informative and representative and coolest places to see of a city. And you can see that probably for many places this might be true. So many of these tall buildings are actually really very interesting. But you can also see that you might miss some really interesting and cool aspects of a city by just always going for the tallest, for the most intense ones. The SRM or PRM way of sightseeing Zurich is very different. So there you would, before you fly into the city, sit down at home and take a Zurich city guidebook. And based on what you previously learned about the city, you would decide, okay, these are the places that for me personally are really sounding very interesting and cool. And then when you are in the city, you would just spend time on specifically visiting what you have defined as interesting before. So this would include, of course, some of the tall buildings, like the Uetliberg from Zurich or the Prime Tower. So there can be the same buildings as already with the shotgun approach, but you can also include the tiny things of Zurich, like a great cheese fondue place or some of the beautiful windows in the churches. So also the more small approaches can be included here, which you might miss by doing shotgun. And the swath or data independent way, acquisition way of sightseeing Zurich is again very different. So what you would do here is you would fly into Zurich and you would photograph everything. You would just go out of the plane and you would start non-stop photographing everything, every house, every person, every street, everything you see. And only when you're back home, again relaxed, hopefully, you would start taking a Zurich guidebook, you would learn about the city where you actually have been, you would look at all the pictures that you made, and then you would make a selection out of what you have taken, and you would decide, okay, these were actually really interesting places, this is what I want to show my friends, and this is what has been cool. Okay, so this is, in a way, in a very simplified version, definitely. Not everything is 100% comparable to mass spectrometry, but it's a very easy way to understand the different approaches of these three different technologies. Are there any more questions to this first part? What is SRM? How does it work? And why is it something useful to do? Yeah?
Sorry, do you mind? Thanks.
Thanks. I just have a question about the settings. I mean, if I'm interested in several proteins or several peptides in the same sample, and you want the quantitative data to be comparable between these two, and do you need to set up the same dwelling time or cycling time for all the peptides or proteins, or you set up optimized for each of them?
Mm-hmm. No, so you're asking if you have several samples that you want to compare, whether you would use different optimal settings for different samples.
Yeah.
And that's definitely not what you do. You use always the same SRM transition settings for every sample to have this comparable. You would always measure the same transitions with the same dwell time, usually with the same collision energy, between all the samples that you want to relatively compare. Mm-hmm. Actually, this is very important because otherwise the intensities cannot be nicely compared.
Yes, but what if the chromatography between the peptides is different?
The what?
I mean the chromatography,
the chromatography.
Yeah.
Yeah. So for example, that's what I said, that you can have a peak with a longer elution than the other one. It's something you could do. So you can, the instruments allows you to use different dwell times or so for certain transitions than others. But usually you take like the average peak width as your standard to define how many. This is what most people do. And for projects where a lot of peptides and proteins are involved, this is the fastest way to go. If you want to have the optimal assay super defined, you can optimize per transition or per peptide these parameters like dwell time, also collision energy, and so on and so forth. But often this is something that you do when you are really interested in having the absolute optimal assay for just very few peptides over many, many samples. So it's a bit of a trade-off what your assay is about. Yeah. But usually the most of the scientific projects is you take the average peak width, this defines your cycle time, and then you apply this constantly to all of your transitions.
Okay, thank you. Yeah.
Okay, so if there are no more questions, I mean, I'm also here until Wednesday. You can anyway at any time come and ask. How do I design a targeted proteomic experiment? So in order to perform now your first own SRM experiment, you need to have three types of information. You need to know what are your target proteins of interest. You need for each protein know what are the best representative peptides that you want to select. And for each peptide that you have selected, you need to know what are the optimal transitions per peptide to choose. And now over the next slides, I will go through these three different types of information and try to give you some guideline or tips and tricks how you would make this selection. Okay, let's start with the proteins. The protein selection, which are your target protein, of course, depends on each project. So actually, there's not much I can tell you. It's just something that comes with each project, and you can define your protein selection either on the literature or on any other experimental data that you have available. What's maybe interesting to consider is the number of targetable proteins depending on the 3 different targeted approaches that you use. So if you are planning to do an SRM experiment, the perfect target protein size that is nice to work with is somewhere in the range between, let's say, 10 and 100 proteins. And the moment when you are significantly above targeting more than 100 proteins, it's very tedious to do an SRM experiment. And then you maybe want to consider to rather go for one of the other techniques. With PRM, you can maybe increase this number a little bit. So in the range of maybe 100, I think the PRM, the champion PRM paper, where most peptides have been targeted in a single run, is in the range of 600 peptides. That's a paper by Bruno Domon, where he measured 600 peptides in just one injection. And if you are really planning to do a proteome-wide targeted analysis where you have thousands of proteins that you want to analyze, then the SWOT or DIA technique is the only choice that you have, that you can use. Okay, once we have selected our target proteins, the next question is which peptides shall we choose for these ones? Because each protein, if we work with a protease like trypsin, typically yields tens to hundreds of different peptides. And the big question is, which ones of those shall we choose? And what people do here in literature is they try to choose peptides that fulfill 2 criteria. First of all, they try to select those peptides per protein that are the best MS flying peptides. So these are the peptides of the protein that give you the highest signal intensity in your SRM or whatever else measurement. Because if you go for these best flying peptides, you can be maximally sensitive. You will be more easy above the noise threshold than when you take low flying peptides. And the second criteria people try to fulfill is peptide uniqueness. So try to select those peptides of your proteins that are really unique for your target proteins, that are not shared with other proteins in your sample, in order to be really selective for this one target protein. So now next question, how do you find what are these best flying peptides? And here you have 2 options, and what most people do, and the best way to do this is to use empirical data. So use data that either is out there and has been measured by other people, or use your own experimental data and see for a certain protein which peptides have been identified. There are several online repositories here, just 4 examples, where people upload mainly their shotgun data usually, and you can go on those websites and you can just, when you have a list of target proteins, you can look on those websites which peptides have been reported for my proteins. If you don't trust the data of other people and you rather just trust yourself, you can do own experimental surveys analysis. So you can perform your own mapping experiments, usually by doing shotgun or discovery proteomics, on your own samples and find out, in my lab, on my samples, I find these peptides for these proteins. To increase the coverage, so the sequence coverage, or to see more proteins, because what can happen is that sometimes you don't see your target proteins in this initial shotgun analysis of your target samples. You can, for example, fractionate your samples of interest by doing any type of peptide or protein fractionation procedure, like isoelectric focusing by off-gel electrophoresis, or by doing strong cation exchange, whatever. Just any type of fractionation that helps you to detect more peptides and more proteins. Or you can in vitro synthesize your protein, so to generate a sample where your target protein is very high abundant, so that you very likely get a lot of peptide hits for this protein. Or a third option that you have, you can select every theoretical peptide per protein. So you can just in silico digest your protein sequence with trypsin, and then you get 10 or 100 or 500 peptides. You make a list of all of them, and in an initial survey analysis, you measure all of those peptides with SRM, and then by the end of this pre-experiment, you will know what are the best peptides in my experiment for this one. However, this is of course only feasible if you have very few target proteins, because otherwise it will take you years to cycle through all theoretical peptides. And then there is a third option. So another option, if you have no empirical data available, if you're working with an organism that has never been studied before, or if your target proteins never show up in any experimental data analysis that you did, you can use prediction algorithms. So there are several prediction algorithms published. Here are 3 examples shown. And what all of these algorithms do is they take the primary protein sequence and then they digest it, and based on the amino acid sequences of the peptides, they try to predict which will be the best flying peptides. And usually these prediction algorithms perform less good than trusting empirical data, so it's really something you should consider as an additional option, but empirical data will usually always be beneficial to trust this one. Yeah, Brandon?
Yeah, there's a new one out from the Macoss lab. There's a new one out from the Macoss lab that's actually an external tool to Skyline now, and a lot of research from Brian Searle. I think there's a published paper showing that these other ones really do not work very well at all, close to random guessing, basically. And his is slightly better than random guessing, but basically, yeah, empirical data is definitely better.
So there's a 4th one to add and to make fat or so.
Called Prego.
Prego, okay. Yeah, so usually empirical data is the way most people try to go, and it often makes sense to spend some time on finding these best peptides empirical than spending a lot of time on measuring something that is bad quality over many samples. Okay, and there's also a scientific expression again for peptides that fulfill these 2 criteria, and this is the term proteotypic peptide. So peptides that are best flying and unique for a target protein is what people call a proteotypic peptide. Maybe you've heard it already in the literature. However, in the last years, a new or another term has been used a lot in the field, and that's called a quantotypic peptide. Has any one of you heard about this term before? Never? Nobody? Yeah? So could you explain what a quantotypic, do you know what it means?
I'll try. It's a peptide. It's a proteotypic peptide that's actually good for quantifying the peptide, the protein.
Perfect. I would have said it exactly the same way. So every quantotypic peptide is also a prototypic peptide, but a quantotypic peptide fulfills some additional criteria that make it optimally suited for protein quantification. And these additional criteria I tried to list here and to very quickly go through them. So one additional criterion that a good quantotypic peptide should fulfill is it should be completely cleaved during digestion from the mother protein. And when you work with trypsin, it is already well known that, for example, miscleaved or partly tryptic peptides, so peptides with not tryptic ends, are not ideal for quantification because you will never be sure how much of this peptide actually is miscleaved or partly tryptic. Again, it's well known that trypsin does a bad job of cleaving these what's called rect ends, usually so KK, RR, RK, KR sequences on either side of the terminus. So if you use a peptide that has these sequences, it's quite likely that you might not get 100% specific, 100% cleavage, but that you get some versions where still KK or RR are at the C terminus. Then it's known that trypsin does a bad job of cleaving peptides that have close to the cleavage site, close to the positively charged arginine or lysine, a negatively charged amino acid. So try to avoid those. And also KP or RP sequences are not ideal for quantification. Usually we always assume that trypsin does not cleave KP or RP, but it has been shown that sometimes it actually does. So again, if you can, for quantification it's nicer to have this sequence not included in your quantotypic peptide. So all these are rules that should try to help you to ensure complete proteolysis of your quantotypic peptides. Another group or another criterion that you can think about when it comes to quantotypic peptides is to avoid peptides that are prone to chemical modifications. So peptides that carry a tryptophan or a methionine are prone to oxidation. Ideally don't have these 2 amino acids in there. NG, QG sequences are prone to deamidation. And N-terminally QE or cysteine, carbamidomethylated cysteines can cyclize. So again, these are all sequences you ideally don't want to have in a quantotypic peptide. And then there is this last criterion, and that's peptides that carry biological modifications, that carry PTMs you don't want, because very often a PTM is not quantitative, right? It's not 100% phosphorylated, your peptide, or you can never be sure about these things. So I think this is actually the most difficult part to take into account, because of course we don't know all the PTMs yet for any organism. But one thing that you can do is you can check online repositories or use prediction tools to figure out whether on your selected peptides maybe some PTMs have been detected before. You can avoid well-known motifs. For example, the NXST glycosylation motif is quite well known, so this you can try to avoid. Or you can avoid the very N and C-terminal peptide sequences. As it's known, they are more prone to degradation, or they are chopped off as signal sequences, and so on and so forth. The hard reality is that all of these criteria here are of course very valid, and they all help you to get better quantitative results if you fulfill them. The problem is, if you fulfill them, you very often do not end up with a single MS detectable peptide per protein. And so what people do is, in reality, they start to relax on some of those criteria to still end up with a peptide they can work with. And there is just one more comment that I want to make here, and that's if the purpose of your analysis is to do relative quantification, so to compare proteins or peptides or PTMs between samples, then especially these first two criteria here, if you are able to control them in a way that you control them that they are reproducible over all the samples you want to compare, these two criteria become less important. Let's say, so if you do not manage to cleave a peptide completely out of the mother protein, but if this is reproducible over all the sample that you want to compare, your relative result will still be correct. Same with if you get methionine oxidation in the same way on all the samples that you compare, your relative quantification will still be correct. So especially to relax maybe on those two criteria, if you do relative quantification, can still be makes you feel less bad, maybe, or that's where I would start to relax on things. However, if you want to do absolute quantification, and we talk about this a lot tomorrow in the lecture, then these things are actually also very important, and it's not so easy to just neglect them. Are there any more questions to quantotypic peptides? No? Good. So now we have selected target proteins, and we have selected proteotypic or quantotypic peptides for them. The last thing we need to select are now the transitions. And again, every peptide that we decided for will produce a series of fragment ions, not just Y and Bs, but also water loss, amine loss. And you need to know, if you would take all of them, you easily end up with 100, 200, 300 transitions per peptide, and we can just not afford to measure so many transitions per peptide. So the question is, which transitions shall I use for my SRM experiment? And the answer is actually very similar to what we have already discussed on peptide level. People do exactly the same. They also try to select the most intense transitions per peptide, the ones that are the highest in the signal, to be really sensitive. And they try to select a transition combination, so a unique ion, unique transition signature, in order to be really selective for one peptide and not to have a mixture of several peptides that can be described by this combination of transitions. And the way how people select it is again the same. Ideally, you use empirical data. So in order to select the most intense transitions, what you need to have is an MS2 spectra somewhere, where from an experiment where you have an MS2 spectra which has been identified as your target peptide, and from this MS2 spectra you can then select the most intense transitions. So you can use any of the repositories that I just showed you on the peptide slide before. If MS2 spectra information is available, you can select those most intense transitions. And especially in recent years, also more and more databases are created that really contain targeted proteomic data. So from those databases, you can directly download transition lists, and if your target peptide is included, just download them and plug them into the mass spectrometer and start measuring them immediately. And also For transition selection, you do not necessarily rely on the data of other people. You can generate your own data by doing measurements where you end up with an MS2 spectra. So you can use shotgun, but you can also do SRM triggered MS2, or you can do a parallel reaction monitoring experiment. All of those generate MS2 spectra that you can search, and once you have identified a peptide, you can take the most intense transitions from there. And people do this survey analysis by using either of this approach, either again on the samples of interest, or they work with synthetic peptides, because then you increase the chance that you will get a good quality MS2 spectra, or they use the in vitro synthesized proteins, or again, your last option, select all transitions per peptide in a survey analysis, measure them all, and after this measurement you will know what are the most intense ones for you to work with. But yeah, again, here usually the people work with a Y-ion series exclusively, and this can only be done for a few peptides but not on large scale. And just one more comment, also for transition selection, algorithms have been described. So also for transition selections, you have algorithms where you provide the peptide sequence and they will tell you most likely Y5, Y7, and Y3 are the most intense ones. But empirical data is always superior. Okay, so in order to wrap up this part, if you want to do an SRM measurement, you need for every peptide that you target what we call this SRM assay, targeted proteomic assay. And this assay contains parameters, information from the whole workflow that you apply. It contains information on what are your target proteins. It contains the information what are the best proteotypic or quantitypic peptides. It contains the information how these peptides then behave on the mass spectrometer. So what is the Q1, the Q3 values that you want to target, and at which collision energy do you want to measure these targets. All of those parameters that I show you here are strictly required. If you don't have them, you cannot start the measurement. Nevertheless, there are two more parameters that we also store in our SRM assays, which are not strictly required, but which are optional. One of this is the peptide retention time. So this is not necessary if you do non-scheduled SRM. However, in the moment when you want to do scheduled SRM, it actually becomes a strictly required parameter as well. And the last parameter that we also store in every assay are the relative transition intensities. So we also store the information which of the selected transitions that we select here is the most intense, what's the second intense, what the relative intensities between those transitions. And this information is not required before the measurement. You don't need it to set up the transition list, but it's very, very useful after the measurement for the data analysis. And you will see this a lot throughout the rest of the week, how you use this for your confidence in identification and how to use this in automated peak picking analysis and so on and so forth. Okay, and all those different parameters, they are stored simply in form of these tables that we looked at already. So every SRM assay is nothing else than a big table. This is how the information is stored and how it's distributed and how you would download it from assay repositories. These assay tables contain always several columns, the protein column, the peptide column. All of those columns here describe how the peptide behaves on the mass spectrometer. Q1, Q3, relative fragment ion intensities. The column of dwell time and collision energy we already discussed. And the last column here is the retention time column. And here Ariel will talk a lot about this afternoon, so I don't want to say too much to this one here. But in this column here, if it's provided in an SRM assay, it makes, of course, not a lot of sense to store a retention time in the unit of minutes or seconds. Because, of course, if I can measure a peptide at 15.5 minutes on my instrument, on your instrument, this can be a very different retention time. So the retention times are always LC setup dependent. So a much better way to store retention time information is to somehow normalize this retention time in a way that you have a unit-free retention time information that you can easily transfer between different LC setups. And this is what we call here this normalized or IRT retention time, and you will learn a lot about this later throughout this day. So generating all these parameters is actually very cost and time intensive, and I always say that on the past on all the SRM projects that I was working, I spent much more time setting up this table than actually measuring this table over many samples. That's just the nice part at the end of the project, and you need to invest much more time, and it makes also sense to invest more time to have the optimal assays right from the beginning. And the good news is that if you once have developed a nice assay that works for you, this can be stored and other people can use this as a starting point for their analysis as well. So by providing assays to these public repositories, or by going to these public repositories, you can make this process of generating SRM assays a community effort. So, and I can just encourage you, if you start doing SRM experiments or SWOT or PRM experiments, really donate your assays to these libraries, to these online repositories, and make it a group effort, a community effort to generate them. And also for you, it can be very helpful if you find your target peptides on one of these websites. Any more questions to how I design a target proteomic experiment? Yeah.
So in SRM, at no point you measure the intensity of the precursor itself. It's always the intensity of the...
Exactly. In an SRM measurement, you have no MS1 intensity information measured.
And question, 2 slides before this one. So the last peptide, so maybe this is not specific experiment. The last peptide that you have selected has at least 3 methionines, and so here we are talking about the non-oxidized form of any of those 3 methionines.
Exactly. So usually you should have here the oxidized form. If you target the oxidized form, it will be another peptide having indicated that this one peptide is oxidized or not, this amino acid is oxidized or not. So this was the non-oxidized version of it.
And you would go rather for it than for them?
You would go for, ideally you would go for all versions of it, the singly oxidized, the doubly, all combinations of it, because it's also, it's a different peptide whether this and this is oxidized or this and this, so you must make combinatorial all versions of it. Ideally avoid the methionines if you can. If you cannot and you want to do a very decent job, you try to target all of them and see which one you can detect, and you would assume that the ones that you are detect are the prevalent ones in the sample. And ideally also, if you do relative quantification, each version should give you the same ratio. The non-oxidized one, the same ratio change between 2 samples than the singly oxidized and the doubly oxidized. There were more questions? Yeah.
So are there any instances of a single transition being used for a peptide where only one was good? I mean, has that ever been made it to the publishing stage or not?
Definitely. So there are definitely papers out that just quantify peptides with a single transition. However, it's not desirable to start like this. So what often happens to me is that I started with 4 transitions or 5, and then maybe just one transition ends up being not interfered and useful for quantification. So what's definitely very helpful is to have several transitions for identification, because that's what we will see soon. It is also an issue of really identifying your peptide, and if you have just a single transition, it's almost impossible to very confidently identify your peptides, unless it's a very high abundant peptide. Then what you use for quantification is again another question, and many people use several for identification and just the best for quantification, so you always must differentiate what the people have really done. But even for quantification, I personally think it's always better to have several transitions. It gives you more data points, more data, more robust data to use several as well. But there are many publications out there that just use a single one. Yeah.
Sorry. So you mentioned several times it's really dependent or important to start also from empirical data, but I mean in our hands it's often very, very much dependent on the background of the sample, whether you will measure or not. And so based on your experience, I mean, is there any guidelines you could to predict the success of an assay? Like, I mean, if people want to measure the protein in serum versus a cell lysate versus something else, I mean, like starting from a discovery experiment, would you say, like, if you never see it on a high-performance discovery instrument, it's not going to work for SRM as well?
You are depressingly right, definitely. So when you're targeting a protein that has never been detected before, or you're working with a PTM peptide that has never been detected before, the chances that you will be able to detect it are definitely low. They are not zero. And if it's a super important project, you might go for it. But yeah, very often what has not been detected in the big screens before, with using extensive fractionation, it is not likely. What I experience very often is that when you're working with very short peptides. So shotgun proteomics is very bad in identifying short peptides because the algorithms expect a certain number of fragment ions, which just doesn't happen when you have very short ones. Very often there I had beautiful SRM traces. Once I had the synthetic peptide, it got me very beautiful results which were never reported by shotgun before. So that was an exception. Maybe that's an exception where it's maybe worth going for a very short one. But nevertheless, we had these big projects ongoing where we ordered thousands and tens of thousands of synthetic peptides from companies, so in a high throughput. And those ones that have never been detected before were also very poor. Then later on, when we had the optimal assay to still see it in the endogenous samples, was the one with the low success rate. Yeah. And you're also right, it completely depends on the background as well. So it can be that somebody in their cell line detects a peptide you don't detect at all in yours, and so on and so forth. Or you have a certain transition interfered that is not interfered in another background. And that's just another reason why it's so important to start with. Always start your experiment with several peptides that when you have outliers or differences, you can just easily get rid of them and still have something else to work with. Same with transitions. Don't use just 2 transitions. If one is interfered or both are interfered, you end up with nothing. When you do your assay optimization with 6 transitions, it's not so painful to kick out 2. So you always need to think first, I need to spend some time to optimize the assay, stay rather broad, and once you know what's optimal in your experiment, it's easy to shrink down then. That's the way I always do it. Yeah?
So do you also use multiple charge states when you look at them?
Mm-hmm. Yeah, so usually I always try to go, if there is a dominant charge state, I go for the dominant charge state. So dominant means like if it's 2 plus is 60 or 70% and the 3 plus is only 20% or 30%, I only go for the higher abundant charge state. If I have 2 charge states that are really 1-to-1, you can also use both. Or you can even make a combination of charge states. So if you want to select 5 transitions for a peptide, you can use 2 in the doubly charged and 3 in the triply charged charge state. Nothing prevents you from doing that. Do you see what I mean? So usually you would go always for the most intense ones. But for most peptides in SRM, the doubly charged version is very often more intense than the triply charged on triple quads, but not always. And usually one charge state is significantly higher than the other. So it's rare cases where you end up with 2 charge states and still would like to work with both of them. Yeah?
Can you pass the microphone?
Thank you. So I just have a question concerning when I'm doing my discovery work in the beginning and I'm looking for transitions on Orbitrap. It's an HCD cell.
Mm-hmm.
And then I do my my SRM on a regular CID cell. Am I going to see huge differences between the 2 instruments?
Maybe I'll just refer to Ariel's talk later on, and HCD mode is already good, the good way to do your discovery. But you're completely right. Different fragmentation mode in discovery, and then will have a huge impact on the intensity.
The only reason I'm saying that is because when I'm looking at triply and doubly charged, fragmentation may be completely different. Yes. So I may have precursors which are really high in triply charged, but the fragmentation looks completely different to what I'm going to see on the Xevo.
And you're right, and there is no way to
actually
solve
predict
this.
Yeah, exactly.
The way to solve this is to make your assays on the instrument where you do the SRM measurement. So this is why people often do this SRM triggered MS2, where you're on the triple quad generating. Then you get the best match or consistency in these things. And it's something you need to be aware of when you come from another instrument.
Thank you.
Good. So then I very quickly go through the last part. We still have 5 minutes?
Yeah, we have a long coffee break.
Okay. Because this is just a nice part to really show you how the data looks like. Well, at least those people who have not seen data yet, SRM data yet. So here's once more a wrap-up of all the different triple quadrupole vendors that are out there. Most people usually work with Q-traps or TSQ instruments are the most popular ones. But there is also Agilent, Waters, Bruker, Shimatsu that provide triple quadrupole mass spectrometers. And this is always the case in industry. With each instrument, there comes the specific software. And in the past, it actually has been literally impossible to directly compare data that came from different vendors, because you were never sure whether the differences that you see are actually really due to the measurement or whether they are due to the software processing afterwards. So when Brendan started his Skyline project, actually we were very happy that this project was designed right from the beginning to be able to work with all the different vendors and all the different formats, and to be able to analyze them all with the same software. So you are now the lucky generation that can benefit from having a software that is really able to work with any type of vendor. On this slide, I would like to summarize 6 important criteria that you can apply to reliably identify a peptide in an SRM measurement. And you will see that these 6 criteria will also come back over and over again for the rest of the week, and they also apply in the same way to PRM data and to swath data. So it's really an important slide, I think, and once you memorize this, everything will be much more easy also with the other techniques. So a first important criterion that you can apply, we already discussed, that's the criterion of coelution. So if you measure 4 transitions, as in this little example, for one peptide, what you would expect, all of them are coeluting in retention time. And in this blue transition, for example, you see this is clearly shifted. The retention time apex is clearly off from the other 3. So this blue transition is very likely due to an interference of another peptide with similar Q1, Q3 masses, or from a contaminant in your sample. So for quantification, you should definitely remove this one transition and just trust your quantification on the other 3 remaining transitions. The second criterion is peak shape. So if you measure transitions of the same peptide, they also should have exactly the same peak shape. So in SRM, if you have traces like this, where you have a weird non-Gaussian peak shape, this is actually reassuring, even more reassuring for you that all of those transitions shown here are coming from the same peptide. And then there is the third criterion, and that's signal intensity. This criterion is a bit... you need to take a bit with care, because but what it means is that when you work with an optimized and validated SRM assay, it's quite likely that the most intense peak that you will see in your chromatograms is your peptide of interest, is your single peptide. However, if you are unlucky and the transitions that you have picked are shared with another peptide in your sample that comes from a protein that is very abundant, and you are targeting a low abundant kinase, it can well be that your target peptide is the small guy here and not the big one. So you must take the signal intensity just as one criterion out of several others that you combine, but you cannot take it exclusively. Is that clear? So it's one additional piece in the puzzle that you can use, and very often the most intense peak is also your target peak, but don't trust it completely only on that. So these 6 criteria you can always apply. For this, you do not need to have any prior knowledge about your peptides. You can just take a transition list that you made up out of your mind, you measure it, and you can look at these 3 criteria. Where do I find chromatograms that coelute, have the same peak shape, and are very intense? Unfortunately, these 3 criteria are not sufficient very often to still really confidently identify a peptide in the sample. And this is why it's important to have additional criteria. And a 4th criteria you can apply is the correlation to a library spectrum. So if you have an identified MS2 spectra coming from whatever source, where you have found your target peptide and you have selected, now let's say here, those 4 transitions, what you would expect is that the intensity that you see in the peak that you think is the peak is somehow reflected also in the intensities that you see in the MS2 spectra. So the Y6 is the most intense, then the Y9, then the Y11, and so on and so forth. if you find this correlation, you're more confident that you're looking at the right peptide. A fifth criterion is correlation and retention time. So if you have prior information about where your peptide likely will elute on your column, you're more confident that you're looking at the right thing. And Skyline actually can predict for you, you will do this and you will see how this works, retention times in your measurement. This will be indicated in this yellow shaded box, and you see here this is the predicted value by Skyline, and when you're close to this predicted value, your confidence increases. And the last and actually the most powerful criterion you can apply is correlation to an isotope labeled reference. So if you have an isotope labeled reference molecule in your sample, what you expect is that your light and your heavy, your endogenous and your reference are perfectly co-eluting and have the same peak shape in terms of chromatography, and you also expect that the relative intensities between your light and your heavy peptide are exactly matching each other, exactly mirroring each other. Okay, and now on the next slides I would like to practice a bit these 6 criteria with you based on some real data. Okay, so let's first have a look at a very nice example. So this is data from myself where I was targeting a protein in a yeast total cell extract using this peptide and using these 5 different transitions. This was actually a non-scheduled SRM run, which you can see from the fact that over the whole gradient of 45 minutes, I ran an LC gradient of 45 minutes, you get data for these 5 different transitions. And what you can see now immediately is that there's just one retention time where all 5 transitions have the same peak shape, are co-eluting, and are by far the most intense peak in this whole chromatogram space here. So by looking at this picture without having any more information needed, I'm very confident this is my peptide, this I will use for quantification over all my samples, I'm done. The problem is that very often it doesn't look so nice. So I think this RPS6A is one of the most abundant proteins in yeast. This is why it looks so nice. Much more often, SRM chromatograms look like this. So here, I was targeting a kinase with those 5 transitions, again, non-scheduled. And now you can see that there were 2 retention times at 16 minutes and at 23 minutes, where you have coelution, where you have peak shape, and where you have similar intensity. What do you think is the right peak, at 16 or at 23 minutes? Which one shall I choose? The one at, maybe we can make a guessing. So who thinks 16 minutes is the correct peak? Raise your hands. Nobody. And who thinks 23 is the correct peak? You're not allowed to not raise your hand. So why do you think it's the 23?
I mean, the most intense peak is the Y9, and I mean, not all the time, but most of the time it's longer, the peptide, you get higher intense peaks. It's not always the case, no?
No, not at all. But
okay. Just my guess.
This I wouldn't agree on. Any other? Why did you use the...
Because the...
Stop. Sorry?
Is it because of the prediction, retention time prediction?
But there is, you don't see any retention time prediction.
Oh, that's
here.
right, yeah. Well, I'm wrong there. Next.
You are a visionary. Anybody, something else? Why, any other idea why to pick the 23 and not the 16, or why to pick the 16 and not the 23? So the answer is that no, you know the slides, Brandon. You're not allowed.
What about the number of transitions?
The number of transitions. I mean, in both cases of the 5 were measured, but you're right. So here the blue transition is not present. So actually that's true. That's one guess that would rather hint to this, where you have all 5 and here you have just 4. But it can happen also in reality that the right peptide has one transition which is just very low because it was already in the MS2 spectra, one of the low ones. So I wouldn't say that this completely rules it out, but it's true. It's nicer. It points more towards this one. But nevertheless, I would never ever publish a paper with these 2 peaks and with having only peak shape, coelution, and intensity as the criteria, because you can really not be sure. So the people who didn't raise their hands were actually right, because you don't know. You can really hardly judge from what I show you here. So what you need is more information. So what can we do? I repeat it, so checking the library spectra. Yeah, so luckily I have a library spectra here for you. So that's the MS2 spectra that I used to actually generate this assay. And you can see that the most intense transition in the MS2 spectra is the Y9. So the Y9 is the purple one. So you can see here the purple one is most intense. Here is only the second most intense one. And Skyline conveys to you this information how well these MS2 spectra fits to those two chromatograms in a very fast and easy way by providing for you dot products. So a dot product is just a mathematical correlation between those intensities and those intensities. It can be between 0 and 1. 1 means perfect correlation, 0 means none. And you can see this has a very good dot product of 0.99 compared to 0.82. So with this additional MS2 spectra information, I'm very confident here we are looking at the right peptide, here not. And I could publish the paper. Yeah?
Just to come back to the missing transition, one thing that you can do visually, even when you don't have a spectrum, and I've done this in lab meetings at the Makas lab, is if somebody puts up something and says, oh, this is my peak, and you look at the transitions, and the selectivity of the transitions is also very important. So if somebody's showing you Y2, Y3, and Y4, and those are the three transitions that are showing up nicely coeluting, and they're missing Y8 and 9, and in this case you're missing Y11, which is your most selective transition, then it's like that's a good indication that, well, you know, I'm not quite so positive about that, because, you know, your most selective transitions, the ones that are most likely not to be in other peptides, are missing. So, but people will get convinced by, like, oh, look, I see coelution by, you know, 3 less selective transitions.
And also goes to what you said, that so the higher molecular weight transitions are more selective, but they don't tend to be more intense. So the connection with intensity, I wouldn't say, but it's definitely true what Brendan said. They are more selective, so it's more likely to find those only in a single peptide than in many others. Okay, so we solved the problem. The next problem is that very often in SRM, if you're targeting low abundant proteins, the situation looks even worse. So this is the trace for this protein here, this protein targeted with this peptide and 4 transitions. What's the right peak? So I show you the MS2 spectra right away. So that was the MS2 spectra from the library, and I show you the dot products of different intense peaks here. And what you see is the dot product doesn't help us here at all. So we have 0.99s and 0.89s and 93 quite frequently. And that's something that you often see when you have an MS2 spectra with similar intensities of many transitions. This is also exactly what you see with noise, right? Noise is similar intensities of many transitions. So very often you have also getting good dot products with very noisy data just picked somewhere. So here the MS2 spectra cannot help us. What can we do? Yeah? What was it? Be brave. Say it. I heard.
In these cases, you can use MS to the 3.
But for that, you would just use, so do an MS to the 3 over the whole gradient as well, which will take a lot of time from you. I mean, it's definitely something you could do, but I think there is something better to do, something more easy, without changing the settings of the measurement. Yeah?
Retention time?
Retention time information.
What about retention time?
You can try to predict retention time to figure out whether you rather need to look in that area or that area. And you will learn how to do that. Yeah, that would be one option. What's another option? The strongest, best option of all?
Using synthetic peptide?
using a synthetic or using an isotope labeled reference. Can be a synthetic peptide, can be a SILAC experiment, whatever. So in this case here, I didn't have a synthetic peptide and I wasn't allowed to order one. So what I did was I prepared a completely labeled yeast extract, which is very easy in yeast. You just need to provide 15N labeled ammonium sulfate to the media, and then you will generate a completely labeled proteome, and I spiked them 1-to-1 and I compared them. And this is the 15N labeled trace, and when you overlay them, there was actually just one retention time where a light and a heavy channel were perfectly coeluting. This is shown here. So at these 11.8 minutes, I got those traces in the light and those in the heavy. These were actually close to a predicted retention time that I got with SSR calc. This is one of the dot products that was very good, 0.99. And the relative intensities, even though this peak, no question, is not looking as nice as the peaks I showed you before, but the relative intensities do match, heavy light do match, retention time does match. All my 6 criteria are fulfilled. So now I'm confident that I'm looking at the right thing and I can do quantification with it. And without having all the 6 criteria, it would have been very vacuum and very difficult to do that. And just to be complete, this is an example of where I was targeting a peptide for a protein, where even though using isotope labeling as a reference, I'm just not able to identify coeluting transitions. So even though SRM is a very sensitive method, it can be that you will not be able to identify anything in there. Don't start integrating stuff like this or stuff like this. You need to have a proper peak still. Okay? So that's all I wanted to show you in some data so that you get a feeling. You will see much more data and much more examples over the week. But these 6 criteria are really, if you understand them, I think you can follow many of the things that will come up. Any more questions to them? Great. One more?
What about pre-fractionating your sample? Could it be done?
Sure. I mean, you can pre-fractionate the whole workflow that you do, whether it's shotgun or SRM, or you can do with any, it's a sample preparation workflow, right, that you're talking about. You can do this with an SRM experiment, as with an PRM, as with a shotgun. And it will help to improve your sensitivity if you, by pre-fractionation, reduce the complexity of every sample that you spike in. Could definitely help, but it makes more work, and you need maybe more injections per sample if you have several fractions, and you need to also consider how reproducible it is. Many ifs, many things to concern.
May I ask a couple of more questions?
Sure.
One would be, I only worked on orbit trap instruments, and I was always told that quads are better on triple quads. And quadrupoles are much better on triple quad instruments.
Quadrupoles are better on triple quad instruments. The quadrupole itself.
Yes, to isolate precursors.
Oh, this is something I wouldn't say. I don't think so. It's just, it's just the quadrupole is what is very good in terms of sensitivity. That's the slide that I showed you. So you're very efficient you have a high transmission efficiency, and you're very good in reducing noise. And this is why the triple quadrupole instruments have this wide dynamic quantification range. And there are also publications that show a very wide dynamic quantification range, also on an orbit trap, on a Q executive. I am not the expert I don't want to compare all of those, but definitely I think SRM is some of the most sensitive techniques you have in hand, and often this is why you can measure the least abundant proteins with SRM, which you not get on an orbit trap in PRM mode or shotgun.
Just a quick question. You showed this.
Well, with PRM mode, probably even. Maybe I don't.
This 4 or 5 orders of magnitude that one can quantify. So it's for, if the peptides is different by 4 orders of magnitude in 2 different samples, this you can reliably quantify with SRM.
Well, we have a whole lecture on that tomorrow.
Okay.
I will come back to this point. Why it will depend on where in the range of linearity you are. Whether you can quantify 4 orders of magnitude difference. But we will discuss this in detail.
And if you have reproducible problems with your LCs, it's too bad.
Mm-hmm. Well, this is always bad.
But if it's reproducible, if it's always tailing...
You have reproducible bad chromatography.
Yeah.
Well, I would assume that this will also not help your sensitivity and your CVs, but if you see that your CVs are happy and you look at the data and you're still convinced you're looking the right thing, but I guess it would still be worse to solve the issue, or at least spend time on solving the issue. And if it doesn't work and if it's reproducible, you will see it by doing replicates, and I mean, it's very important to do your homework and figure out what's your CV, what's your reproducibility rate, and then it can look okay. Yeah, so I have only the take-home messages. Shall I? Well, but on the other hand, I think you also have, you will get the slides, so this is just the summary once more of what I told you and the outlook that so far we have only talked about identification and not at all about quantification, which is of course the main purpose of doing SRM, but this will all come tomorrow. Great, so thanks a lot for your attention, and if you have more questions, I'm here, and just come and see me.